Okay, good morning. Let's start. So, for today, as I said, I want to make this a bit more practical. I'm not quite sure yet how this will work out. I guess we can work a bit on the computer, and my idea would be that I occasionally make break, or quite more often make breaks so you can try something out on your own computer. Now today this will probably be a little bit slow because everybody needs to get the data and the software and so on, but let's see how it goes. The idea is a bit that often math is more easily understood if you work with the data yourself and experiment a bit. And let's start with something simple, and let's just try to understand this analysis that we did so quickly with this black box Sora tool on Monday, and do this slowly and see what's happening there. And I sort of prepared a little bit of simple analysis steps where we can stop at various places and discuss a bit about what's going on there. So let me see what I have here. I've written it all up here, but I will get later into the details. So this is sort of my cheat sheet. I will put it online afterwards, but for now you better look at my RStudio. Let's start with who in the audience has never worked with R so far? Okay, but you've worked with other programming languages, so we will probably, I'll add a few extra remarks about the peculiarities of R so that you get a feeling for it. So what I want to start with is with this same example data set that we had last time, and maybe while I'm talking you can already download it. I quickly try to find the link for us. I think I've put a link on, or did I put a link here on this one? Yes, no? PBMC, here I put the link to this, but we can also go through the actual company web page, and I think they should have it somewhere here. Products, Chromium single cell, software instrument resources, and here they have all their different example data sets that they have. Oh, we've added a lot of new stuff since I did this last time. And what we now have, actually, if we want to, we can simply try to do something. As I told you, this what I used is some very old data set from the very beginning of it. So maybe if we are brave, we can just decide to start with one of the updated data sets. Let's stick to PBMCs. Stained with total TBNC gem, chromium 10X, Ringle cells, 3', melanoma, so we have here lots of example data sets we can work with. So to make my life easy, I start with the one we had last time, but of course maybe it would have been better if I used something here with 10 million cells instead of with 3 million cells. But I'm not quite brave enough to now switch data set and work with another data set than what I've prepared. But maybe you want to, and then you can compare whether the stuff I demonstrate here also works with one of the newer data sets. When you download this data set, you see here it comes in a zip file, and how did I no, sorry, I'm still a slightly here temp PBMC3K, and I unpacked it, and hey, didn't I unpack it? Okay, then I have to download it again. Download it here. Okay, here PBMC3K, CD PBMC3K tar XVF downloads. How was it? What did I just download? That's why it doesn't work. I'm on the wrong computer. CD tim / here, I've unpacked this thing, and you see it has here this with directory called filtered matrices. It's called filtered because I told you initially, if you get the raw data from the machine, it gives you a count matrix with several 100,000 columns. And if you then look at these several 100,000 columns, you will notice that most of them have very low column sums. And the way people like to display that is plot, which probably we should make ourselves, but I show it, they call this a knee plot. And it looks like this. Let's say here, col sum, so the x-axis is the total number of read counts, and it's usually written in a way that you do, other way around. Y-axis is the counts per cell. And what you then have is something like maybe so I have a double logarithmic scale here, and the way how you read this thing is some kind of a cumulative density plot on a log scale. So you see it falls off monotonously, and the way how you do that is you read it off like this and say, the 10,000 largest columns, so the 10,000 columns with the largest column sum have all above 300 columns, or the 100 top columns have all about 10,000 column sum. And this double logarithmic thing allows you to quite quickly read off that you can see up to 1,000 columns have sort of a large number of things, and then it steeply drops off. And they call this here a knee. And then you just make a cut here and say, okay, these are probably the real cells, and these are empty droplets. Now, where do these empty droplets come from? I probably have to explain a little bit about how this 10x machine works. So it has this 10x video. Let's see if there's a nice dropseq microfluidics videos. Oh, no, that wasn't it. This one. Here you see how this works, how you get these single cells put into a droplet. So through this here is oil, which comes in here. So the whole thing is maybe a micrometer across. So this here is oil coming in through these two entries, and this here is water coming in, or water solution comes through these three entries. You see from the right comes these big things that are the cells. So the cells are entered as a single cell suspension coming from the right in this thing, and these little silica beads that I told you about that you buy, and where each silica bead has a different DNA barcode coated all over it. These are these little points which come here from the top. I'm not quite sure why it comes from both sides, but and as you can see, this is water, and here comes oil from that side, which pinches off water in oil droplets. The funny thing about this emulsion, because you know, if you go to microscopic fluid dynamics, everything changes. All the Reynolds numbers are very different, and suddenly the dynamic looks quite unintuitive. So this is all laminar flow, despite all these complicated shapes. And as you can see, the droplets are all exactly the same size and shape and so on. And what we now have is that these droplets, hopefully, they should contain one cell and one bead. But here, this would be a proper droplet with one cell and one bead. What's also in the droplet is all kinds of chemicals, especially a detergent which lyses the cell membrane, so that the cell membrane opens up and the RNA is released into our reaction volume, so the droplet. And the droplet also contains material for reverse transcription. So in order to help the RNA to attach to this, produce this, maybe I have to draw this. So the bead, these beads, they contain these oligos, and the oligos are attached to the bead, and at the end are lots of copies of T's. And then comes here our random barcode. And many copies, always with the same barcodes, and another one which is a random barcode, which is different between each of them. That's something we go. And then what now happens, what I haven't told you on Monday, is when the cell makes a copy of a protein coding gene, one of these transcripts, the last step is that it attaches a chain of A's to this transcript. So if we have such an mRNA molecule, it has a chain of A's, which base pair with a T. So this thing sort of sits here and complements to it. And if when the polymerase comes, what it will do is, and here is now the actual end of the actual gene's transcript. And there's also polymerase in the droplet. So all the polymerase enzymes, they are added here to this solution. So that they come in there. So the moment the cell membrane is lysed and the RNA is released, the poly A ends of all the messenger RNA will attach to the poly T's at the end of all these little oligos on the bead. And now the polymerase, that's also in solution, can elongate, as the biologists say. So produce a copy here which is attached to it. And then you have your bead, and the bead has copies of all the transcripts. Each transcript attaches somewhere, and we have a copy, or not of all of them, of maybe 5 or 10% of them. And the nice thing is, each of these copies now consists of a transcript of the bead, the poly T stretch, and then this barcode which identifies the bead. So we can put all the beads together afterwards and get these and sequence them. Now, what happens? But as you can see, this is a stochastic process. We just randomly put in cells and beads and so on, and just try to get the concentration of beads and cells just right, that on average each droplet contains one cell and one bead. And the number of cells which we actually have in droplets is simply Poisson distributed. And you want to make this Poisson distribution such that the number of, and the rate parameter of the Poisson distribution, of course, depends on how concentrated your single cell solution is, how many cells you have per milliliter of fluid. And now you have to have a trade-off here. If you want to get many cells on your beads, then you add a lot of cells so that you get a lot of data. But when the probability becomes high that more than one cell will end up in a droplet, and and then we have a problem because this this bead will contain RNA from a mixture of 2 or 3 cells. What will also happen is we don't want to waste cells by being in a droplet without any beads, so we put in way more beads than cells, and that then has the effect that there's quite a lot of droplets which are empty. They contain a bead, but they contain no cells. But while we prepared the whole experiment, some of the cells might already have been broken before we fill it into this machine. So this here is the hard piece of this machine that this company 10X sells. And before we fill it into the machine, a couple of cells might already have broken up and released the RNA, which is now in solution in this whole fluid. So even with empty droplets like this one, which contains only a bead, contain a little bit of RNA, so that afterwards we will find beads which not contain a few thousand RNA molecules, but only a few dozen RNA molecules. And that's this lower part. And as this happens much more, so we get something like a few thousand real molecules and a few tens of thousands of empty, what we call empty droplets, which didn't contain a cell but did contain a bead. And the first thing you do when you analyze this data, once you have your count matrix, you calculate the column sums, and then you make this plot. Making this plot is a nice little computer exercise to think about it. I tell you the solution. You take all your column sums, you sort them in descending order, and then you plot the column sum value on the y-axis against the order number of this thing, like first, second, third, fourth highest number. So you make a table where the left table is just numbered from 1 to 1,000,000, and the right column has all the column sums, so the total number of read counts per cell, and in descending order. And when you plot this table on a log-log plot, then you get this. And that's the most convenient way to then see these two regimes of actual cells and empty droplets. And hopefully here the transition is deep enough that you can sort of send the thing. The data that they put us here as our example data with 3,000 cells contain exactly 3,000 columns, so they cut here at 3,000 and didn't even remove this. And usually if you work with this 10x data, you get two data matrices, one with all the columns and one where the small columns have already been cut off. So these are this typically kind of small things that you spend a lot of time when working with real data. I point this out maybe for the mathematicians who are, I imagine, used to the fact that if you get real data, it's probably prepared, but in bioinformatics, we say 90% of the biometrician's work is data wrangling, as we call it, preparing our data and bringing it in the shape of actually preparing it for analysis. So, this is what we have now here. We have this matrix here with these that we get here, which is called the filtered matrix, because it's only the columns with big enough column sum that we actually consider them real. Here it says that it was aligned against the Human Genome Reference version 19. And there's now the actual matrix, and the matrix is provided, as I said, it's a sparse matrix, and it's provided in a format which is called the matrix market format. It has an old history. It was introduced, I think, by Boeing for some fluid dynamics simulations, or by NASA some 50 years ago, and it's mainly used by MATLAB. And it's very simple format. Here you have this matrix market, and then you have here a column which tells you the dimension of the matrix, number of rows, number of columns, and number of non-zero entries. And then it tells you here, okay, rows, this row and this column has the entry 1, this row and this column has the entry 29, and everything else is 0. And of course, then we have a raw matrix, but we need to know which matrix, which row is which gene, and which column is which barcode, and this is why there's these two text files here, the text file genes.tsv, which just gives you for each row of a matrix, just in order, first row, second row, and so on, the database ID in the ensemble genome database for each gene and the name of a gene. And in the same way, there's the other thing here called barcodes, which contains you all the barcodes which were read from this beads. The barcodes have also a little other thing. The barcodes are designed the way that, as you can see, they are quite long. So we have, how many are these? Maybe 12 or 15. So there is 4 to the 12 or 4 to the 15 possibilities, or maybe a few 100 million possible combinations. In fact, in our experiment, we will have only 1 million. We will only have up to maybe a few 100,000 beats of this. As I said, most of them are empty. And that means for any 2 beats which we find, the edit distance, so the number of bases you have to change to get from one beat to the other has a certain size. So we imagine that these beats live in a space like A, C, G, T, 2V, let's say, I think it's 15, or maybe somebody can count. And in the space of all possible strings taken from this here, we can set a distance matrix. And the distance matrix we simply define as the distance between 2 arbitrary strings is the number of places where the strings have different value. And if we do this, we can easily imagine that it's rather unlikely that within our 1 million beats taken from this 100 million possible combinations that we find 2 beats whose sequence have an edit distance of only 1. And that's useful because it can easily happen that the sequencing machine misreads a sequence and then adds a new column for a single molecule rather than adding it to the molecule where it is. So we do a correction step at the beginning where we take all the barcodes we find and try to find all instances of barcodes being having a distance of only 1, and then we say, okay, this barcode we have seen 300 times, and then this barcode we have seen only twice, and it differs from the first barcode by only 1 change, so let's imagine that it's the same, and let's correct the barcode by replacing this change. And you can imagine this is a nice combinatorial problem of discrete mathematics, where you say, I have these, in an event, it's a vector space of matrices. I have this distance matrix, and I want to find an efficient way of finding for each barcode whether it should belong to the whether it should stand in itself and be its own column, or whether it should be added to another column by finding all pairs of barcodes which have an added distance of only 1. And we have to do this in an efficient manner because, as you say, we have 10 million, we get maybe 10 million of these barcodes out there, of these beats out there, most of which have only appear once or twice. And if we would simply compare each barcode with every other barcode, we would have make 10 million squared comparison operation, which takes too long. So you have to think about how do you bring this up from the squared scaling to an n log n scaling. And this is an issue they had when writing their 10x software, which makes this count matrix, because the count matrix we are looking at here now has already been corrected. So the barcodes, which are these barcodes, which are the column headers, they also include barcodes which have been shifted by undoing potential read errors. But if you want a nice little sort of computer science puzzle, try to think about how would you program this barcode correction algorithm. Can you come up with something which doesn't take square scaling? So, but in any way, this is our simple file format. One big table with row index, column index, and value there for all the non-zero elements, and then the labels for the rows and the columns. In R, there is R has its own sparse matrix thing, which is defined in the matrix library, which is one of the standard R libraries. And in this matrix library, there's this sparse matrix function, which allows you to construct such sparse matrices. So you give the sparse matrix a vector with all the row index, with all the column indexes, and with all the values. You also give them a value pair with the dimensions, so just 2 numbers, rows and column. And if you want to, you can also give it the list of rows, the list of labels for the rows and for the columns. And so what we have to do is simply read in these 3 files, get our 5 vectors with 3 columns from the first files and the 2 label vectors from the other files, put them to a sparse matrix and do this thing. The other possibility that we can do is that we take the Seurer function, which already has a function called read10x, which does exactly what I've just described. You give it a data directory, in my case, temp pbmc3k filter gene matrix, and here are the 3 files, and with read10x, just expect that there are 3 files in this directory with these 3 names that we see here, and we'll read them in and put them into this sparse matrix construction thing. And when we do this, we get our count matrix. I call it CM. If you don't have Seurer installed, you could, of course, just call sparse matrix manually, but then you have to first write the 3 read table commands to read in all the data and to read in the 3 files and get the vectors out of them. And then the next thing, I have now a look in this matrix. We've already seen it last time. It has this representation of all the zeros represented as dot. But in R, we can have a look at how this actually looks internally by looking at its internal structure. And as you can see, this thing has an index vector with row indices for the 22868, what is this, 22 million, 2.2 million non-zero values. It has here the actual 2.20 non-zero values. And it has this pointer thing here, which is not the column indices, because there are 3 different customary ways how to store a sparse matrix. And I quickly show it to you because we will need this occasionally. And it's nicely shown in Wikipedia, where we have these 3 storage things. The storage in the file was what they call what people typically call the coordinate list, or people also call it the triplet format. But there's also the compressed sparse row, which is shorter. As you can see here, we have in the files, we had 3 vectors of this long length, but here the third of the 3 vectors is much shorter. And this is achieved the following way. Let's imagine you have this matrix here, where you have sparse entries. Basically, each row typically does contain 1 or 2 rows elements. And then the way how this works is you have one vector which tells you the actual non-zero values taken row by row, so 5, 8, 3, 6. When the column indices tell you which columns they are, column 0, column 1, column 2, column 1. And then you have the row index, which tells you at which element in this V thing a new row starts. The row 0 starts with value 0. The row 1 starts with value 1. The row 2 starts with this value. The row 3 starts with this value, and then it ends. Now, this is actually, here's a better example where we have multiple values per row. So you can see the row index now tells you the values in the 2 long vectors. The values 0 and 1 are the first row. The values 2 and 3 are the second row. 4, 5, 6 are the third row. 7 is the fourth row, and 8 is the end. So this is a storage which saves space by not repeating the row index for each of them, but only telling us where it changes. That's called a compressed sparse row format. Whenever you work with these sparse matrices, so you have these big matrices with lots of zeros, you typically will spend a lot of time thinking about which of these 3 things are most suitable for your task. And suitability not only depends on what is the best storage format which saves most time, but also what allows you to most efficiently carry out the operations that you want. So a typical exercise here would be think about how you would implement a matrix multiplication of 2 such sparse matrices, and which of these 2 formats is more suitable to make a matrix multiplication. You can already see this here is very good if we want to go through a matrix row by row, because you know where one row ends and the next row starts. But if you want to go through column by column, you jump back and forth. Now for a matrix multiplication, you always multiply row by column. So ideally would want to have the left operand in row sparse and the right operand in column sparse. So column sparse just means that the role of column and row index is reversed. And as you can see here, our data set here has decided to do it the same way as using the long index. So this is the long index. The column index is here the row index. So this is a row sparse matrix. But luckily, we can load a library called sparse matrix stats, which contains already pre-written code to do this efficiently, calculate matrix multiplications, row and column variances, and row and column mean for such sparse matrices. So, now let me have a quick look of what I had prepared for you. So this is how this count matrix here works. And yeah, that's what I already told you last time. We can look at the dimension. We can look at the row names and column names because our count matrix here has the rows and columns labeled. And when we can look at the column sums, for example, col sums Cm gives me now for each cell or for each beat the number of elements that we get there, and I can sort this in a descending order. Decreasing gleich true. And now I have them here in the decreasing order, and now I can plot this on the x-axis. I just put the number 1 to n row, no, n col count matrix. So for the non-R people, n col is simply the number of columns of the matrix. 1 to n col is just all the numbers from 1 to 2700, which I put now on the x-axis. And this here is the column sum in decreasing order. And now if I do this, then I get this plot that I showed here, but it doesn't look like on my blackboard because I have forgotten to logarithmize the x and y axis. And if I do that, then you can see here exactly how this works. Here, these are the actual cells with between 20,000 and 2,000 things, and then it rapidly falls off. And here is then sort of the foot where it goes straight out to the right again, because what we see here is only the first part of the plot because we have already cut away the rest. I like this plot because it's an example for a pretty non-trivial plot, which is very easy to make. It takes a while to get your head around the fact that it really makes sense to sort data and to plot an index on a logarithmic scale, but as you can see, it's helpful to see these sharp decreases. Next thing I can show you here is, let's look at a histogram of this col sums, Cm. And if I make an ordinary histogram, I don't see much. Well, here I do see a little bit, but it's more helpful for many of these things if I make the histogram of a decadic logarithm, because then the whole thing looks much more symmetric, and we can easily read off where the mode is, namely here, remember 10 to the 3.5 is something like 3,000 at around 2,000. Now, first job is, as all these cells differ so much in how many total reads they have, we should now ask whether we can actually, whether it makes sense to compare actual read counts, or to compare, or whether we should put it in relation to these column sums. Let's look, for example, at this CD3E. You might remember this from Monday, where it was this gene which marks the T cell receptor, which marks T cell with one of our different things. And we've seen here that most of the cells don't have any genes for the T cell receptors because they are not T cells, but some have. And if I plot this, then you can now see here the non-T cells, and here some T cells stick out. But it does help to plot this against the column sums on the x-axis. By doing it like this. And then we can now, for example, easily see that these are clearly T cells, because even though the column sums are small, the number is high, so this is a large fraction of all the reads fall onto this, while here, this is maybe not as convincing. So it would make a lot of sense to normalize the data by dividing each count value by the corresponding column sum. And what we then get, if I do this, I get not the total counts, but instead I get a fraction. I get for each gene what fraction of all the reads that have been sequenced in a given cell, what fraction of all these reads have fallen onto this specific cell. Now, if we want to do that, we take the count matrix and divide them by the column sums. Now, and after we do this, the column sums of this normalized matrix should be 1, which it isn't. That's the point where I now stop for a moment and let you puzzle about what's wrong here. How do I properly normalize my count matrix such that for each cell we replace the counts by fractions, and hence the sum over all the the column sums add up to 1. Okay, it's a simple thing, but it's early in the morning. So who knows where the mistake here lies in this thing? Yeah, exactly. In R, we have this rule that whenever you divide some multi-valued thing, like a vector of matrix, by another multi-valued structure, it does something which it calls recycling. It divides the first element of this by the first element of that, the second by this by the second of that, until it runs out of values in one, and then it starts at the beginning again. And that does what we, and here what this does is, if it goes through whenever R goes through through the entries of a matrix, it goes row by row. So then, of course, it divides the first element through the first column for sum, the second element in the, sorry, that would work. Now it goes through column by column, very much in contrast to what all other programming languages do, which goes through it row by row. So if R would go through the columns of the matrix while going through the column things. The way to fix this is very simple. I just transpose my matrix. And then I divide by my column sums, and then I transpose back. And now it works and all the column sums are 1. So I call this fractions because it tells me the fractions of all the things, and because this is so confusing with the transpose, and I can never remember whether the slash operator operates on columns or on rows, I have a habit, whenever I do something, I double check the columns afterwards to see whether really the columns add up to 1 and not the rows or something else. So, this is how I get to these things. The next thing I want to do now is to look at the rows and column variances. What I've done here is, here I made this plot where each dot is a gene, so a row in my fraction matrix, and I plot the average of this gene over all the cells against the variance over all the cells to get this plot. For the people who are used to R, this is probably obvious. For the others, I have to tell you that in R there is a function called row_vars and col_vars, which calculates these means and vars by it. So let's do this. Row_vars, fracs, ifs, now. Row means, you see there's, for whatever reason, there's a row means, and the row means 2, so R is sometimes a bit disorganized. And there's also a function row_vars, fracs. Actually, when this was new, when I did this for the first time, when I ran row_vars on my matrix, it told me that row_vars doesn't know how to deal with sparse matrices. So I had to change it into a dense matrix and or write my own row_vars function. And that's what I did. And it's also an exercise which you can do if you want to sharpen your numeric skills. Given this column sparse row format, how would you best calculate row variances of such a matrix given by that? And you can already see how this works. You have to look through your row index and say, okay, I go through the values 0 to 1, 2 to 3, 4 to 6, and so on. And for each of them, I calculate the mean over the values, and then I calculate the squared mean distances, and then I have to divide them, of course, not by the number of actual values, but by the number of values, including the zeros I skipped over. The difficulty, if you do this in practice, is that you have that R is a 1-based format, so all the matrices and vectors are labeled starting from 1. Especially the computer scientists will know why this is bad and why this causes you a lot of headaches. So you keep adding and subtracting a 1, but let's not do this at the moment. So, luckily by now somebody has added this functionality and put it in the sparse matrix stats function. And if I now do this, I get here this matrix, and as you can see, it seems that the variance has a noticeable dependence on the mean. You see how the higher the mean is over these functions, the higher is the variance with some sort of baseline and some stuff that sticks out. If I want to find out such a dependence and wonder, is this a linear dependence? Is this a quadratic dependence? What's the best, easiest way to find out what kind of dependence this here might be? Anybody got a good idea? That's now a question for the physicists in the audience, because it's one of these standard little physics hacks that you learn in your experimental practicals. You have a scatter plot and XY plot, and you want to know what kind of power law the relationship is. So you have something like, why do I keep losing my things? You have something where you suspect that the y-axis might be some, yeah. Exactly. So I have I suspect that this is a power law, something like a to the a times x to the b. And if I take the log on both sides, then of course I get log y is log a plus b log x. So any power law will turn into a line in a log-log plot, and the slope of a line tells us the exponent. So let's tell this thing to make our axes logarithmically. And as you can see, there is a clear line here. Question is, what is the slope of this line? I could now start trying to read this off, or I put some grids into it, and actually what I've done when I prepared this is I made this plot that I've just shown you here, and then I put these two lines in here, just manually using these segments here. And you see the first line here, I go up on the decadic scale from -6 to -2 on the x-axis, and from -10 to the -6 on the y-axis, so 4 decadic units. So this is slope 1, that's this one, and this is slope 2. And as you can see, our variance seems to have a slope 2 on the left, and this then turns into slope 1. So our suspicion is that the variance of these things is some sum of two parts. One is proportional to the mean, and the other is proportional to the mean squared. And we will talk, I think, in the next lecture about what actually the logical or mathematical reason might be to expect exactly this variance mean dependence. To understand this variance mean dependence in a nice way, it helps to look at the coefficient of variation. So coefficient of variation is what we call the standard deviation over the mean. So the coefficient of variation is this typical thing when you say this value sort of this value has an uncertainty of 10%. This is what we call a logarithmic noise, because it's always 10% of the current value. If you have twice the value, then the absolute uncertainty will be twice as high, but the relative one stays the same. So with relative uncertainty, that's the coefficient of variation. And looking at this coefficient of variation can be helpful. So what I now do is I look at the coefficients of variation squared. And so V over mu squared is A times 1 over mu plus b. So we imagine that there's two components. One is some kind of multiplicative noise, which will later turn out to be the biological part of the noise. If you have a couple of genes, a couple of cells which are supposedly in the same cell state, then we can expect that the expected value of the expression of a gene should be some value, and the scattering of different cells of the same type around this expected value will be a certain percentage of the actual value. This is why we say this biological noise is multiplicative in the sense that the CV is constant, or that the standard deviation is proportional to the mean, and hence the variance proportional to the mean squared. While this here is something which comes, so what's the first thing you think of when you see variance being proportional to the mean? What process gives you a variance that's proportional to the mean? Because usually, as I just said, we would expect variance to be proportional to the mean squared, because when the standard, yeah. Nope, no goes. A distribution which is famously, because the Gauss distribution is a two-parameter distribution, you specify mean and variance separately, so you can do what you want. And typically, in many Gauss distributions, you specify the standard deviation to be proportional to your mean. So that's a typical case of multiplicative noise. Now, the thing where you have a variance being proportional to the mean is the Poisson distribution. In the Poisson distribution, the variance is proportional to the mean, and we expect Poisson to play a role here, because, as I told you on Monday, we have all these many molecules in the cell, and we only sequence a small part of them. So we select randomly a few percent of the whole, which means that this is a binomial process with a low probability, and the binomial distribution becomes the Poisson distribution in the limit of small success probability. By the way, I guess you all had some stochastics courses, so you remember these, at least might remember these little tidbits about Poisson and binomial and so on. If not, maybe try to remind yourself of it, because we'll need it a lot. So this is what we have here, this thing, and in order to see this in our plot, what I now want to do is divide the variances by the mean squared. This is what I did on the next one. No, I divided it on the mean here, because if I divide the variance on the mean, then the part, so here's the variance divided by the mean, and if I divide the variance only by the mean, then I get A plus B mu. And so this plot here is now the variance divided by the mean, and we can now here see the Poisson noise part. A is now constant, and the biological part goes above. And that's what we see here. We can see that all these bulk of genes where the Poisson noise dominates, they sit here on this line. So you see this here is variance by the mean. And on the top here, that seems to be the cells where we have appreciable biological variance in top of what the Poisson noise generates. Now you might wonder, why is our Poisson noise not equal to the mean, but only proportional to the mean? That's simply because we made this normalization where we divided each cell by where we divided each value by the total values for this cell. And we can do a bit of math, and maybe we'll do this in one of the next lectures to find out what the actual expectation is of this Poisson noise, of this value, and it turns out it's the average of the reciprocals of these normalization factors by which we've divided, and that gives me this value here, which gives the orange line, which, as you can see, goes straight through the middle. So in this way, we are now able to put our genes into two categories. Those genes here, these genes here are the genes where the variance is dominated by the Poisson part. And this is it. So this is even if the cell, the expression of this gene would be exactly the same over all the cells, we would still see a variation due to Poisson. So even if B would be 0, we would still have that part. And the reason why this is of interest is we want to make use of our data, and we have this huge matrix. We want to find out where the interesting stuff is in this big data matrix. And obviously, the interesting part are we want to find the genes whose expression varies from cell to cell, because these genes will be able to tell us something about how the different types of cells in our sample differ from each other. While genes, and this we can say this only if that noise term dominates. If this noise term dominates, then of course we can't say anything interesting about the gene, because all the variation is just the technical issue that we have only a limited sampling efficiency. So these are the genes which either have no differences between the cells because they are exactly the same cell in the same expression strength in any kind of cell, no matter what the cell does, or they are overall so weakly expressed that we simply don't get enough data to see differences between cells. And that's the two things that you see here. Here we have the genes where we have only a little bit of data. And here we have the genes which probably could show differences but don't. So this is what the biologists call housekeeping genes. Genes that all cells have the same need for because they just support basic functions that every cell needs, and so they are expressed in the same strength in all the cells. While this here, on top here, these are the cells that are of interest for us. That's the cells we really, the genes we really want to look at, because these are the genes that give us information about what different kinds of samples we have in our cells. So, and for data use, I keep these genes here. So here you see what I've now done is I've taken, as we've just argued, we should divide the row vars of these fractions, so the variances for each gene, by the means in order to find those which have an interesting large enough value of B, so it's interesting biological variation. And if I sort all of them, then I get on top of it the most useful genes, and at the end of the list we use less genes which don't contain information for us. So I take the first 1,000 of these genes, take their names, and call this the list. These are the top 1,000 genes. We'll need them in a moment. 1,000 is of course an arbitrary value, but what might make more sense is to somehow put a dividing line here along this area and catch, in order to catch this cloud and say, let's take all these genes. But I once counted it, this is about 1,000, so if I take the 1,000, I'll get about these ones here. You can actually also plot this here by saying colon equals, if else, see, row names, fracs, percent in highly variable genes, comma, red, otherwise black. And now you see here which genes these are. And to get the same plot that we had before, I divide by the row means. And yep, so that's now where I've cut, or here about. So now the next thing we do is we make a logarithmic transformation. As you can see here, because the next thing we want to do is we wanted to define a distance matrix between our cells. And if we define a distance matrix between our cells, the easiest way it would be to say each cell is a vector of count, so a column in our matrix, and we just take the Euclidean matrix, the Euclidean distance between any two such columns. But if we take such a difference, then the sum of all these differences, so the Euclidean distance, will be dominated by the differences of these highly expressed genes, because of course we add up differences on vastly different scales. There will be differences here at the scale of 10 to the minus 4 between maybe this gene in two different cells, and differences here at the scale of 10 to the minus 8. And obviously, the value of the Euclidean difference between any pair of cells will be dominated by these few genes, and all the other genes down there won't have a chance to won't have any chance to influence our difference at all. So, but this is not what we want. We want the distance to reflect differences in all the genes. And there, it now makes sense to transform our data in a way that all these genes are sort of on the same footing. We might do it like this, dividing by the row means and then taking this thing. The other thing which people like more is that they say, as we've already said, this B here tells us how much the gene scatters. And if we divide and ideally, if we now ignore the Poisson noise and say the variance is given by B times mu squared, we want to make a transformation such that the variance is equal for all the genes, independent of mu, that all the genes have this variance B and just B. So we need a transformation which takes our data and transforms it such that this multiplicative noise turns into additive noise. Multiplicative noise means the standard deviation is proportional to the mean. Additive noise means the standard deviation is constant and is independent of the mean. And how do we change and if we have a family of random variables where the variance of x is proportional to the expectation of x, and we want to transform them into a transformed variable f on x, such that the variance of f on x is no longer proportional to the expectation but becomes constant. What transformation might this be? Which changes multiplicative noise in additive noise. Yeah, exactly. We take the logarithm because, of course, multiplicative noise means there is some noise part which is multiplied to it. When we take the logarithm, it's this thing added to it. So the logarithm turns this multiplicative noise in additive noise. And in biology, basically, whenever you look at data, the first thing you do is logarithmize for this reason, because in biology, you always have this multiplicative noise. My personal opinion why this is the case, I want to share. You know, usually we will also notice later that a lot of stuff in biology is not normally distributed, but log normally distributed. You all know the central limit theorem. When you have a phenomenon, a random variable, which is influenced by many different random variables, when this summary variable will be normally distributed. People like to say that the reason why body height is normally distributed is that the height of our body, how tall we grow, depends on such a large multitude of factors, something like our genes, and not just one gene, but thousands of different genes. Each of them makes us maybe increases or decreases the expectation value of our height by a few millimeters. Also, lots of little things about our upbringing. Did we get enough food as a child? Were we breastfed or bottle fed? All these things, probably each of them has a small influence on our overall height, and our actual height, which we reach as an adult, will be the sum of all these independent factors. And because it's the sum of many independent factors, according to central limit theorem, it should be normally distributed. If you think about it closely, you realize actually it can't be normally distributed because the normal distribution has support in the negative. Turns out that it makes much more sense to assume that stuff in biology is log normally distributed, because most of the individual factors in biology, they don't add up, they multiply. And this is in the end just a basic fact for chemistry. You all remember the mass action law, or Massenwirkungsgesetz, for those of you who went to school in Germany, which tells you that the reaction rate of any chemical reaction is the product of the concentrations of the adducts. And in biology, everything which all our body is in the end the effect of biochemical reactions, which use up reagents, which are again the product of biochemical reactions. So all the things, including the height and so on, are the result of an incredibly long chain of reactions, and all their efficiencies have to be multiplied according to the rules of chemistry. So of course the result is the product of a huge number of individual factors, and therefore we expect that most of the summary statistics describing an organism will be log normally distributed, because it's a product, and hence the central limit theorem doesn't apply to the variable itself, but to its logarithm. Yeah. But maybe I shouldn't have used the word, because it's probably not the proper word here. But you know, whenever you have data, actually summary statistics is a stupid word. Statistic is the right word. To do this tangent, whenever you have data and you try to summarize this data, you use methods to summarize all your data into a single number, for example, and this is called a statistic. So for example, a mean is a statistic. A variance is a statistic. The median is a statistic. So I used the term in the wrong thing, because the height of an individual body is not a statistic. A statistic is all the ways how you can summarize an aspect of a distribution into a single number. And a big part of statistics, the science, you see the point in English. In Germany, we always say statistic for both. In English, we make a difference. We use the singular word, a statistic, for a number which we calculate to summarize data. And statistics is the science to reason about these statistics. And and of course, a lot of these things in statistics is reasoning about them. That's exactly what we've just done. We've talked about if we want to describe something like human height, how can we describe this distribution? We came to the conclusion, or I just claimed that there's good reason to believe that height is log normally distributed, and the log normal distribution is described by two parameters, the log mean and the log variance, or the mean and variance of the logarithm of the values. And so we say that the human height can be well described by just taking two statistics. And most of statistics boils down to this question of how many different statistics do we have to take to get a good description of a data set, and how precise can we estimate them. Okay, but back, we want to take the logarithm of this data for the reasons just taken, in order to make sure that our data here, which seems to have this multiplicative noise, becomes an additive noise, so that each gene has a decent chance to contribute to the overall distance. And that's what I've now done here. And what I could do is now I take my fractions. Now, but what will happen now is, frax is what I called it. We get a big matrix, and basically everything in this matrix is negative. It's a minus infinity, because you remember it's a sparse matrix, most values are 0. And the logarithm of 0 is minus infinite, so that doesn't help much. Ideally, now we do a hack, which is actually a bit hard to mathematically justify. And because we want to do it properly, we will have to come back to this. But what people do in practice is they just add a small number to this. They decide, let's just take this thing and shift all the values by a little bit to the right in order to avoid the zeros. Now all the zeros have been turned into 10 to the minus 4, and then if I take the logarithm to basis 10, then my zeros become minus 4, and everything which is actually expressed becomes a value larger than minus 4. The upshot of this is, if you look at the logarithm, the proper logarithm looks like this. And now we have just moved this logarithm a little bit by adding this plus 1. We have effectively changed the logarithm to that one. And because it is the logarithm, if we have large numbers, this doesn't make much of a difference. If I have a gene which sits here, which has an expression of 10 to the minus 2, if I take the logarithm of 10 to the minus 2, or the logarithm of 10 to the minus 2 plus 10 to the minus 4, that doesn't make a difference. Only for the weak genes here, this little clutch here will distort our data. And the weak genes, they probably won't help us much anyway, because as you can see, they have not enough variance to actually contain information. Those which do contain information here, they seem to be at least at the value above 10 to the minus 5 in the mean over all of them. The individual values are probably okay. So why did I choose 10 to the minus 4? One reason is because that's what the Sura authors chose when they wrote their package some 5 years ago. The reason why they choose 10 to the minus 4 is that they thought most of our cells have around 2,000 counts. So 2,000 counts, which means the smallest fraction that we see is 1 over 2,000. Something like, and 1 over 2,000, if we add 10 to the minus 4 to this, and just always so bad at 1 over 2,000 is, you see, 5 times 10 to the minus 4, and if I add now 1 times 10 to the minus 4, this changes from 5 to 6 times 10 to the minus 4. So the distortion seems to be rather manageable if we say we add a value which is small even compared to a single read count. And that's why they say, okay, let's take the average column value, which is in our case 2,000. Let's take the reciprocal of this, 1 over 2,000. Let's maybe take not this, but a little bit less, maybe not 1 over 2,000, but 1 over 10,000 to take a fifth of this. And that is sort of a heuristic reason why they say they add 10 to the minus 4. Then they noticed one more little issue. What was my 10 to the minus 4? So what I now would like to do is log 10 fractions plus 1e minus 4. Now I have another issue, and this is a purely technical issue. Remember, fractions is a sparse matrix. Most of its entries are 0. If I now do this calculation, then as we've just said, all these zeros will turn into minus 4. That's annoying. We really would like the zeros to be zeros because all the software is meant to know that a sparse matrix is characterized by most of the values being 0. Technically, we could explain it to them. Now most of the values are minus 4, so wherever you don't have a value stored, assume that it's minus 4, but that is annoying. So let's shift this back by adding plus 4 to shift to make sure that the zeros stay zeros. And that's the, you can see that, and that's the transformation we really do. We take the logarithm in order to turn the multiplicative noise into additive noise. We add 10 to the minus 4 to solve our issue with the zeros. This turns the zeros into minus 4s. Now we shift our whole scale to by adding plus 4 in order to make sure that the zeros stay 0. Now instead of adding plus 4, I can also say I add plus log 10 of 10 to the 4. I can multiply this into this thing here, and then I get here fracs times 1e4 plus 1. You can see that's the same. Now I have multiplied the fraction by 10 to the 4 and added 1, and now I get this, that a 0 stays a 0 and everything else is a little bit higher. there's one more issue here now. If I do this, then our sparse matrix will still turn into a dense matrix. Why does it do that? Because it will have to do this calculation in order. It first multiplies the fractions by 10 to the 4. Zeros stay zeros. When it adds 1, zeros turn into 1. This can't be stored in the sparse format. So R will have to transform our matrix internally for a moment from a sparse to a dense matrix. It will blow up in memory, and if we do this with big data set, our computer will run out of memory. When we take the log 10 and the minus 4s turn back into zeros because we have done it this way, or the 1s turn back into zeros because the logarithm of 1 is 0. But now we've already moved out of the sparse to the dense matrix, and we would have to tell R now convert this back into the sparse matrix, but intermediately it was a dense matrix, and that was completely superfluous. It would have been much better to tell R do this in one go. And because this is such a common problem, R has a special function which is called log1p, which means logarithm plus 1, and it does exactly this. It takes the matrix, it takes the data, adds plus 1, it takes the logarithm, but it does this in one go, so that the zeros stay zeros, and you can, and the sparse, R knows that if it applies this in a sparse matrix, the zeros stay zero. So one issue for log1p uses a natural logarithm, and before we had a decadic logarithm. When I prepared this yesterday, I wondered now, what does Seurer actually do? Does it use a natural or a decadic logarithm? I actually can't remember, we have to look it up. Maybe it might make sense to take this here and multiply it by log 10 in order to switch the basis back from the natural to the decadic logarithm. The difficulty here is that for the life of me, I never remember whether I have to multiply or divide by log 10. I always have to rederive this logarithm basic changes. Maybe other people here are more able, or you might know this issue. That one can, whenever you have a half-life, you remember I have to divide it by ln2 to get the decay constant out of a half-life, or to have to multiply it, and you have to try. So don't ask me whether it's a 0 or a star or a slash here. But this is what we now do to get our normalized logarithmatized fractions. I call this alfrax. Now I simply stay on the natural logarithm to keep my things easy. And now I can calculate distances. So let's take what distance? Let's take a random cell. 1, 3, this cell here. Let's take the vector for this cell. And let's subtract it from all the other cells. So now I subtract the column 1, 3, 5 from every column of the matrix. And square them because I want to calculate a Euclidean distance. And now I have in each column the square distance component-wise. And if I add them up, I should get the distance of the cell number 1, 3, 5 to all other cells. Now here I get nervous again because I used a column and I made this operation where I have made a binary operation between a matrix and a vector, and we had this before, and last time we messed up columns and rows, and maybe we did this again. So what I now will do is, just to be on the safe side, I will check what is the distance between cell 1, 3, 5 to itself, and yes, it's 0, so obviously I did it correctly. And it's really subtracted this vector from the columns and not from the rows. But you can see this thing here is the distance of matrix 1 through 5 to every other matrix. And I make a histogram of that. And as you can see, this is how it looks like. Maybe I should make this with an arbitrary cell. Cell equals sample n cal expressions, comma 1, I think. N call expression is the number of cells. No, n call alfrax, that's what I call it. And comma 1, and sample should give me one random number picked sample int, that's how it's called. One random number picked out of this. And if I do this, and then I take my histogram here, I see, comma, main / cell. I see now the distance of cell 341 to all other cells. And here we might be a little bit disappointed. We've seen last time that we have 3 different types of cells, B cells, T cells, and monocytes. And so we should expect that if my randomly picked cell is a T cell, it should be close to the other T cells, and it should be far away from all the other cells. So it should be bimodal. One thing for the other cells of the same type, and a further away peak for the cells of the other types. But it isn't bimodal. Actually, no matter what cell I take, it all looks more or less the same. The mean is always around 3,500, and the standard deviation is also about the same. And there's a good reason for that. Namely, what we've done is we've added up all these numbers. And again, most of the components in our Euclidean distance, they are dominated by this A here. And they are just random noise. And the differences are more just difference between 2 Poisson distributed random variables, or somehow normalized if Poisson distributed in log. Whatever their exact distribution is, there's a lot of randomness in it. And all these randomness we add up. And all these sum of different random things, which we all add up, or we square them and add them up, what do we get if we have a lot of random variables, which we square and then add up? If we don't square them, before we add them up, we get a normal distribution according to central limits theorem. And if you do square them, we get a chi-square distribution. But yeah, actually here it is it a, anyway, you can imagine that the distance from every cell to every other cell is sort of pretty random because it's dominated by all these little random things, and the actual part that we're interested in, that part gets drowned out by the noise. And we need to find a way to amplify that part and diminish this part. Now, because we are getting to the end of our thing, and also my laptop is running out of thing, I show you this in my prepared lecture. What we now do is, so we log transform it. Here we have our distances. We do a PCA. And what we will do next time is to talk a bit more about why actually the PCA does what it does here. But for now, I take a PCA and I take a package called IRLBA, that stands for Iterated Reweighted Lancor Speed Diagonalization Algorithm. Because you can imagine if you want to do a PCA of a big matrix, means you know PCA involves an eigendecomposition, and making an eigendecomposition of a huge matrix is a non-trivial thing. In undergrad or in your first semester, you probably learned about making an eigendecomposition by writing the matrix minus lambda times the identity matrix. Then you took the determinant of that, and then you got the characteristic polynomial, and you found the roots of the characteristic polynomial to find the eigenvalues, and then you got the eigenvectors from that. But we don't really want to write down the characteristic polynomial of a 1000 by 1000 matrix, because it's a degree 1000 polynomial, and finding its roots will probably be terribly numerically unstable and not probably fraught. But finding eigendecompositions of big matrices is something which occurs in all kinds of application of numerics, and it's a really important problem. So when computers became a sort of mainstream in the 60s and 70s, this was one of the most of an important research area of mathematics, and a lot of idea came up. And we'll talk next time about how these things work internally. But one important thing was thought up by some guy called Lankos, some Hungarian person. Actually, his algorithm never worked, but it stimulated ideas of how to fix it. for example, by making an iterated bidiagonalization of its reweighted, which then gives this IRBA. And this thing now has an eigendecomposition algorithm, which also gives rise to a PCA algorithm, and now we'll run this on L-Franks. Again, we have to transpose because, yeah, otherwise it's to get it the right way. And the specific thing about these things, they don't give you all eigenvalues. They only give you the 20 strongest eigenvalues plus the vectors. So I tell it how many I want. I say I want the 20 largest eigenvalues, largest in the absolute value. And the calculation time is linear in the number of eigenvalues that we do here. And I also want it to center my matrix before it does it. So by center, I mean subtract the mean of each gene, of each row, and it should also scale them. So for each gene, it should divide by its standard deviation, so that afterwards, all the genes have mean 0 and standard deviation 1. That makes sure that each gene has, so the centering is important to make the PCA, otherwise it doesn't work. And the scaling makes sure that each gene has equal chance of influencing the PCA. So, all this PCA. Now that it is a non-trivial calculation, which takes a moment, but should be done in a second. Let me also find in is this the right one? No, it's not. So, usually whenever it takes so long, it means that I messed something up and took the I see what I've done wrong. Even now, there are still too many genes. And we remember we said before that most of our genes are actually non-informative because we have this big bottom here of 10,000 points, which actually are only Poisson distributed. So let's subset our matrix to only contain the rows for these top 1,000 genes. And you remember I put them into the this variable HVG, which is the list of these top 1,000 genes. And if I do that, then my PCA runs reasonably fast. That's also the reason why I used 1,000, because this is, I think, quadratic or maybe even cubic in the number of rows. So by my experience, if you use 1,000 or 2,000, it's okay. And if it's 10,000, it takes too long, or it takes several minutes, and I don't want to wait so long. We can also later try and find out, and this is maybe an interesting exercise to check out, does this really make a difference? What we now get is we get a transformed data, and we'll talk about this in more detail next time, where each cell has now is now described not by a vector of 10,000 genes, by a vector of 20 principal components. Just to make this clear, the rows of these things, they are the cell names. So the cell names are the col names of my expression matrix. And then I also have a rotation matrix. You remember a PCA is a basis change, which rotates our original basis, where each coordinate direction, each dimension corresponds to a gene, into a dimension where each dimension is the principal component. And we make a rotation into this new basis, and then we keep only the first 20 components. And this is why there's also a rotation matrix in here, and the row names of this rotation matrix, they are the genes. So that we see that we can look up there the so-called loadings, which tell us for each principal components which gene names are in there. So these are the no, that's not all of them, that's only the highly variable genes. So, and using this, I can now redo my experiment here with the with the distances. Instead of taking the big matrix with the 10,000 components per cell, I may take the small matrix with the only 20 components per cell. This time this thing is transposed again, so I have to put it that way, and I have to say row sums, and I have to transform this here. I hope I've done this right. No, I haven't, and I show it to you here. Distances, square sum, distances, distances, distances. We're probably about to finish anyway. So, yeah, this the PCAX minus PCX cell comma, no T here, but a row sum here, and I've forgotten my square root in the end for the Euclidean distance. It doesn't work. Oh, now it works. So, and now it works better. Now we often get bimodality. These are the cells which have the same type as my randomly picked cells, and these are the other cells. So we see we now are able to actually find biological signal. It's still not ideally, but we are getting somewhere. So this is how far I wanted to get today. So from the mathematics, this was now maybe not that challenging. We didn't do anything more complicated than doing a bit of division and a bit of logarithm, but we looked at a lot of concepts of how to think about these things. How is our noise determined, so on. Probably we opened more questions when we solved. For example, I still owe you an explanation of why we actually think that the variance has this form and what these two things really mean. And what we should do is set up a model, a data generation model, so sort of some idea how we would simulate our time data. And that's something I want to show you in the next or over next lecture, to give us a feeling of what actually happens in a cell if we describe it mathematically. The other thing I want to do is just to give you a teaser, is I've here still from last year have a nice explanation about why the PCI actually does what we want it to do, namely give us improve our signal and reduce our noise. And we'll come to this, and I want to go through this, but there's a little issue with, we'll have to do this in the week after next, because next Wednesday is 1st of May, so it's a holiday. And on Monday, I unfortunately can't give a lecture because I spent actually the whole week making an introduction to bioinformatics to the new master students of molecular biosciences. So I'll be busy teaching the biologists and probably telling them something very different. So I can't do the lecture next Monday. Wednesday is holiday, so I see you all on Monday in the week after. Yeah, about the format. Now this time I mainly worked on my own computer, but maybe you can now make sure till Wednesday in a week that you have all this software and all this data and so installed. Maybe I also say a bit on Monday, so that when we can do a bit more hands-on, that you actually try to figure out a bit of codes yourself. I didn't want to do this now the first time because waiting for everybody to figure out which way around to transpose the matrix to get the column sums directly is something everybody has to go through once just to get a hang of it, but maybe not while everybody else is waiting. Yeah.
Should we also use R or
in a way you can probably do it as you wish. Personally, I keep switching back between R and Python in my own work. The reason is, as